The male reproductive system. The male reproductive system consists of a set of gland-sand ducts that enable the production, storage and transport of the genetic material contained in male gametes, culminating in the ejaculation of sperm. Semen consists of a fluid, seminal fluid, that contains sperm. This device has two main functions: exocrine function, production of sperm and seminal fluid; endocrine function, production of androgen hormones, testosterone and testosterone. The male reproductive system consists of the main organs as well as the accessory organs. The main organs are: the penis consists of two corpora cavernosa and a corpus spongiosum through which the urethra passes. The two testicles, enclosed within the scrotum. The epididymis. The excretory ducts, urethra and vas deferens. The accessory organs are: the seminal vesicles, Cowper's glands, the prostate. The testicles. These are two oval-shaped glands located at the base of the thighs, weighing around 20 g and measuring 5 cm in length. They are located in the testicles that form the scrotum. Each testicle has 3 layers which, from the inside out, are: the inner lining is acerous membrane, the vaginal. It originates from the abdominal peritoneum. It consists of 2 layers: a visceral layer, against the testis, and apparietal layer, against the tunica media. Between these 2 layers lies a virtual cavity, vaginal cavity. One may observe a hydrocele, pathological fluid between the 2 layers, or a hematocele, presence of blood between the 2 layers. The middle layer, fibrous, it covers the parietal lamina. The outer layer, comprising the skin, it is thin and pigmented. It contains hair and sebaceous glands. In the embryo, the testicles are located in the lumbar region, near the kidneys. As the fetus develops, the testicles descend, emerging from the abdomen through an opening, the inguinal canal. They draw their vessels and a peritoneal diverticulum into this migration, surrounding themselves with it. Abnormalities in testicular migration lead to conditions known as testicular ectopia. In a sagittal section, the testicle appears as follows. It is surrounded by the albuginia, a fibrous, inextensible connective tissue capsule, a thickened portion of which at the superior pole forms Himer's body, starting from the center and radiating outwards. Thin connective tissue septa testis, delimit the testicular lobules, 200 to 300 per testicle. Each lobule contains, within the interstitial tissue, a cluster of seminiferous tubules, 1 to 4 per lobule. The seminiferous tubules of each lobule merge into a straight tube, 1 mm long, which continues into the rete testis, a dense network of canaliculi of irregular diameter, connected to the epididymis. The vas deferens and rete testis are embedded in Himer's body. Epididymis. It consists of 3 parts. Its thick head covers the upper pole of the testicle. Its body follows its external shape. In short, its tail ends directly in the vas deferens. The epididymis contains 2 types of ducts. Efferent cones. These are 12 to 15 tubes originating from the rete testis, which emerge from the hilum of the testis and join the epididymal duct at a right angle. The epididymal duct, a long duct, 6 cm, coiled upon itself and continuing into the vas deferens. Testicular lobule. The testicular parenchyma comprises the seminiferous tubules, the interstitial tissue, vas deferens. In cross-section, a seminiferous tubule appears to consist of a. the proper membrane, a thin outer connective tissue membrane containing contractile cells. B. the germinal epithelium. This is a very distinctive type of epithelium, which is 2/3 as thick as the tube's radius and comprises two types of cells: Sertoli cells, germline cells. Sertoli cells. In optical microscopy, they appear as large cells, L equals 70 m, W equals 20 m, with a complex shape, arranged at intervals of 30 to 40 m along the seminiferous tubules, like pillars. Their basal pole is attached to the lamina propria, and their apical pole reaches the surface of the epithelium. They are also connected to one another at the ends of their lateral extensions by tight junctions, thereby forming a genuine impermeable barrier, the hematotesticular barrier. This creates two distinct compartments: a peripheral basal compartment, an adluminal compartment, communicating with the lumen of the tube. In electron microscopy, the following features are noted: abundance of smooth endoplasmic reticulum, well-developed Golgi apparatus, secretory activity with the presence of numerous lysosomes, phagocytic activity. Germ-line cells. Arranged between the Sertoli cells in more or less regular layers extending from the periphery towards the center of the seminiferous tubule, they represent the successive stages of spermatogenesis, namely, spermatogonial stem cells, primary spermatocytes, secondary spermatocytes, 2, spermatids. Interstitial tissue. A very thin connective tissue, it contains a. the blood vessels and nerves of the testicle, b. clusters of interstitial cells, or Leydig cells. These cells display the ultrastructural features of steroid-secreting endocrine glandular cells, abundant smooth endoplasmic reticulum, and mitochondria with cristae. In fact, Leydig cells are responsible for the secretion of androgens. Spermatogenesis. It encompasses the successive stages that lead from the original cells, germ cells, to the formation of mature sperm. It lasts between 60 and 74 days. It is permanent and continues from puberty onwards. Sex cells gametocele are the only cells in the body that have just 23 chromosomes. The cell has an elongated head, which contains the nucleus. This cell contains all the genetic information carried by DNA. It is topped by a cap, the acrosome, which contains enzymes that play a role in the fertilization process. The cell that contains mitochondria. The tip consists of a flagella made of contractile substances that enable them to move. Its structure is designed to facilitate its movement and to enable it to locate and penetrate the egg. The endocrine function of the testis. The conductor of testicular function is GnRH, gonadotropin-releasing hormone. It is thanks to the pulsatile release of this hormone by neurons in the hypothalamus, which increases significantly during puberty, that testicular function develops. In fact, GnRH triggers the pituitary gland to release two hormones, FSH and LH. In the testicles, these hormones have the following effects. FSH. The FSH enables the development of Sertoli cells and spermatogenesis, the exocrine function of the testis, the secretion of sperm. FSH binds to membrane receptors on Sertoli cells and plays a threefold role. It stimulates spermatogenesis via the Sertoli cell cytoplasm. It stimulates the production of ABP, androgen-binding protein. Finally, it triggers the secretion of inhibin, a hormone that exerts negative feedback on FSH secretion, either on hypothalamic neurons by reducing the secretion of GnRH, or directly on the gonadotroph cells of the pituitary. The LH ensures the proliferation of Leydig cells and the secretion of testosterone, the endocrine function of the testis. Most of the testosterone enters the Sertoli cell cytoplasm, where it binds to ABP to regulate the development of the seminal epithelium and the proper functioning of the genital tract. Seminal fluid. Free testosterone enters the bloodstream and has two effects: a beneficial effect on the genital tract and associated glands, and negative feedback on LH secretion, either. indirectly via hypothalamic neurons, or directly via pituitary gonadotropic cells. The effects of testosterone. Effects on spermatogenesis. It promotes spermatogenesis. A significant testosterone deficiency leads to infertility. Factors affecting the development of the male reproductive organs. Interventions targeting male secondary sexual characteristics. Development of the hair system. Development of muscle and bone mass. Distribution of adipose tissue. An increase in the pitch of the voice due to the development of the larynx. Effects on metabolism. The extratesticular seminal ducts. 1. The vas deferens, 40 cm long. They travel along the inner surface of the epididymis, through the scrotum, and then ascend towards the inguinal canal, which forms the central component of the spermatic cord that contains all the blood vessels and nerves leading to the testicles. The vas deferens enters the abdomen via the inguinal canal. This duct remains within the peritoneal cavity and ends in a dilation known as the ampulla. This ampulla acts as a reservoir for sperm between ejaculations. 2. The ejaculatory ducts. 2 very short channels, 2.5 centimeters. They are entirely contained within the thickness of the prostate. They originate at the junction of the ampulla and the seminal vesicle and empty into the prostatic urethra. This junction of the spermatic and urinary tracts is located below the smooth muscle sphincter of the urethra. During ejaculation, the sphincter prevents semen from flowing back into the bladder and also prevents urine from being passed. 3. The urethra, 16 centimeters long. The urethra carries urine and semen. The urethra is divided into the prostatic urethra and the membranous urethra before ending at the penis. The penis, or the penis, which consists of 3 parts, the root, is fixed and attaches to the ischiopubic rummy. The body, the moving part. The glans, a conical protrusion containing the urethral opening. It is covered by a mobile skin and mucous membrane, the foreskin. The penis consists of erectile tissue, the 2 corpora cavernosa and the corpus spongiosum. They are surrounded by tissue rich in muscle and elastic fibers. The corpora cavernosa are attached to the ischiopubic rummy. They join together and envelop 2/3 of the corpus spongiosum. The corpus spongiosum, located in the midline, 15 centimeters long, traversed by the urethra. Accessory glands. Seminal vesicles located above the prostate. They are 2.5 centimeters long and 1.5 centimeters wide. They produce most of the seminal fluid, in which the sperm swim, as well as fructose. The prostate located beneath the bladder, surrounding the urethra, through which the ejaculatory ducts pass, 2.5 centimeters long, 4 centimeters wide, 3 centimeters high. Composed of several small glandular sacs, surrounded by connective tissue and smooth muscle fibers, each with its own excretory duct. Produces one of the components of seminal fluid. Cowper's glands. These are 2 pea-sized lobes that open into the urethra at the exit of the prostate. They secrete a lubricant to facilitate the transport of sperm. Sperm. It is released with every ejaculation and consists of sperm suspended in seminal fluid. Features. Volume, 2 to 5 milliliters per ejaculation. It is a viscous, white, opaque, flaky liquid. pH, 7.2 to 7.8. Count, 40 to 250 million sperm per mm cubed. Semen is considered fertile when there is a sufficient number of sperm, and when the size, motility, and morphology of the sperm are normal. The test used to assess sperm quality is called a semen analysis.